Hello everyone who study physiology with us, and today we start to study the brain, and we have term brain stem and cerebellum. For today a lot of work and video links, and as usual we have the part in practical book, our lab works, and let's start. We will return to this slide that shows us the structure of the whole nerve system and what we have already learned, and today we will begin to study the functions of the brain. And the brain is a complex organ. It alone takes about 30% of all our resources and is the driving force of over 90% of our body. The mass and average about 2 kilos, and of course very different structure in animals. For each animal, each part of the brain has a special structure, but in general the function is the same, and in the brain there are gray matter and nerve centers. In general, gray matter is on the cortex, and nuclei inside the white matter. White matter, the processes of neurons, they are predominantly covered by myelin. Everything is similar to the structure of the spinal cord, but we have gray matter in the middle of the organ in the spinal cord. In the brain, the gray matter on the surface of the brain, and here we see the gray matter is about 40% of the brain and contains most of the brain's neuronal cells, bodies, and it's just to process for information sources. And finally we have only this gray matter fully develops once a person reaches her 20s, and of course white matter, about 60% of the brain, and made up of bundles which connect various gray matter areas, allows communication to and from the gray matter areas and between the gray matter and the other parts of the body, and develops through the 20s and peaks in the middle age. And inside the white matter we can find the basal ganglia, and I even leave the link here. It is the site for animals anatomy, for humans anatomy too exactly, but this page for animal anatomy, vet anatomy. If you want, you just can to find this information. And let's start to study the methods of brain research. It's very interesting, but how did scientists know what capabilities and abilities of brain? And before considering the function of each part of the brain, you need to find out what method we use for learn, and then here you see commonly used techniques, and for animals too, of course. Very interesting. And of course you can use brain imaging such as X-rays, magnetic resonance imaging, positron emission tomography, and here I will leave the links if you want to know more about it. And of course how we see the electroencephalography too, we have the links for it, but now let's consider rather complicated questions with this division of the brain into different parts, different departments. And how you see here, for understanding the names, let's look at the embryonic development of the brain here on the next slide. Let's start. Here we see the diagram depicting the major subdivisions of the embryonic spinal cord. because the spinal cord is the primary place for development of the brain. And here we see the development of all brain structures and their function too. Here we see the table, and here we see the spinal cord and brain, and we see the three main parts of brain we have: rhombencephalon or hindbrain, midbrain, and forebrain. And today we start to study the brain stem, and you see the much place in the brain. We have so much structures here. Today we will discuss about hindbrain and midbrain. And you see in the hindbrain we can find two big places, mencephalon and metencephalon. And today we will discuss about pons, medulla oblongata, cerebellum, and midbrain too. And what we have here, the function with the region, and we will move from the spinal cord and up. And first place where we'll stop, it will be medulla oblongata, this place green color. And today we will study all structures and functions of the brain stem. You saw how the brain is built differently in different animals too. We will take the model of the human brain and the brain of higher animals, because only these animals and humans has all structures and very important parts for understanding how works the brain. And today let's start to study this place. You saw how the brain is built, and the three distinct parts of the brain stem are colored in this sagittal section of a human brain. The brain stem is the posterior part of the brain that connects the cerebellum, here the gray, with the spinal cord, cerebellum. And in the human brain, the brain stem is composed of the midbrain, the pons, and the medulla oblongata, and sometimes the diencephalon is included in the brain stem too. The brain stem is very small, making up around only 3% of the brain's total weight for humans, of course. It has the critical roles of regulating cardiac and respiratory function, helping to control heart rate and breathing rate. And other roles include the regulation of the central nervous system and the body sleeping cycle. It's also the prime importance in the conduction of motor and sensory pathways from the rest of the brain to the body, and from the body back to the brain. It is almost similar like a spinal cord. These pathways include the corticospinal tracts, motor function, the dorsal column medial pathways, which gives information about touch, vibration, and proprioception, and the spinothalamic tracts with the pain, temperature, itches, clue, touch, and even it also provides the main motor and sensory information. Here we see some nerves supplied to the face and neck via the cranial nerves, and 10 pairs of cranial nerves come from the brain stem. In general, we need to know understand the function with two large brain structures. Here we see the red colored table with two large brain structures. This is the hindbrain and the midbrain. And we will move from the spinal cord, and here the first section is the hindbrain. And this part of the brain includes the medulla oblongata, at first. And the Pons Varoli, the scientist who researched the department and the cerebellum, like a little brain, includes to the brain stem because has all ways for connection. At first, medulla, its first place, the primary role of the medulla is regulation involuntary life functions such as breathing, swallowing, and heart rate as part to the brain stem. It also helps transfer neural messengers to and from the brain and spinal cord. It's located at the junction of the spinal cord and brain, and here we see the photo for human's brain, and here is the cerebellum and the pons varoli and part of medulla, and here just we don't see this, but here was the spinal cord, and what we see here is just the medulla oblongata and the part of the structure from the spinal cord, and here we see the general brain stem, and we see the cranial nerves, of course, and the function for cranial nerves. As you know from this part, in medulla oblongata we have some of them, and we will discuss about it now. Medulla also separates the nerves and control muscle movement, and the nerves that control certain sensation like a touch, temperature or pain. And we have already seen this slide. Here we see the function of the cranial nerves. And medulla oblongata function in general, we have this, of course it's have the part in gray matter and white matter, and we have in general conductive function, a reflectory function, and in general integrative function. In addition, the reticular formation is located in the medulla oblongata, and thanks to this structure, vegetative, somatic senses of taste, auditory and vestibular reflexes are realized. As the conductive function of medulla oblongata is realized by the white matter and conducting tracts from the spinal cord. Do you remember these tracts? And some of them change the side of the spinal cord and move in another side of the brain. And the reflexive function is realized by the nuclei. Nuclei inside the brain stem. You see part of this nuclei here, and here you see the part of gray matter and some nucleus for voluntary and primary involuntary reflexes. So with these autonomic nerve centers, and here are very important nuclei of autonomic reflexes, vital reflexes, cardiovascular center and part of the respiratory center, the center of salivation. Here are the centers of protective reflexes of sneezing, blinking, hiccups, coughing and yawn, lacrimation, and centers of postural reflexes. Do you remember we studied these reflexes when we studied the functions of spinal cord? But some of them, especially for higher level animals, have the centers in medulla oblongata, and even in the cortex, of course, too. So in general we can say that in mammals there are almost no purely spinal reflexes. They all have brain control, and this table shows the levels of control. Now we see the reflexes, the stimuli of each zone. And for tonic, you see tonic reflexes, tonic neck reflexes, and even for some more composite reflexes. With these reflexes, the stimuli of which come from the vestibular apparatus and neck receptors, and we have seen this practical work in the practice of spinal cord reflexes in animals. And do you remember the rabbit? And let's now not forget about these posture reflexes. A special place has the nucleus of daters. Its lateral vestibular nucleus is the second name, and the lateral vestibular spinal tract areas from the lateral and inferior vestibular nuclei located in the floor of the fourth ventriculum within the medulla oblongata, and their fibers project topographically to all levels of the ipsilateral spinal cord, and cells in the anteropostural areas of the lateral nucleus project to the cervical cord, whereas cells in the posteroccaudal regions project to the limbosacral cord. And on each segment of the spinal cord, this tract heaves of collaterals, and thus ensuring that different muscle groups will be coordinated during postural control by extraction and excitatory influence of extensor and flexor muscles, motor neurons, and the coordinated action neurons on providing postural stabilization is not completely understood. And however, if a person begins tilting to the right, ipsilateral fibers elicit extension of the left axial and limb musculature, and consequently the right extensor muscle are inhibited. This action stabilizes the body's center of gravity and preserve upright posture. And the Purkinje cells are the special cells for inhibition, signaling with the help to the neurotransmitter gamma-aminobutyric acid, special transmitter for inhibition. And these neurons we call like inhibitory neurons. The only output neurons in the cerebral cortex constantly control the nucleus of daters, and by inhibiting and activity of the body's extensor muscle, and in turn Purkinje cells are under the control of the red nucleus of the midbrain, and we will discuss about this a little bit less. Normally we have already seen postural reflexes in the rabbit in a previous practice, and many animals has the postural reflexes are primitively developed. In the injury of this part of the brain destroys the connection between the medulla oblongata and the higher divisions. We will see decerebral rigidity, and you see this picture for cats, this one, and for humans too. All extensor muscles are activated in a maximum, as seen in the example. All extensor are activated, and even this body too. This pose indicates brain stem damage, specifically damage below the level of the red nucleus. A red nucleus will be here, and the datus nucleus here. So it is the place where the damage has happened. So next part of the brain, it is the pons varolii. So we discussed about medulla oblongata and the medulla oblongata function, and start to study the function of the pons. The pons, sometimes pons varolii, is a structure in the middle of the brainstem. Its location is inferior to the midbrain and superior to the medulla oblongata. The pons has two divisions, the ventral pons and the tegmentum, but it is different from the tegmentum of the midbrain. And the ventral pons is home to the pontine nuclei, structures that are responsible for the coordination of movement. And this nuclei travel from the pons across the midline and form the middle cerebral pedunculus as they make their way to the cerebellum. And the tegmentum of the pons is considered to be the older region of the pons, evolutionary, and meaning that this structure has present in the ancestors of humans and other vertebrates that have a pons as a part of their nerve system. But for not all animals, the pons is the special part. Sometimes this part is just a medulla oblongata. The tegmentum forms a part of the reticular formation and network of nerves that extend from the medulla oblongata and connect to the spinal cord and thalamic region. And here we see the function of the pons, at first conductive, reflex function, and integrative function too. And one primary function of the pons is the brain is to serve as a pathways for sensory information between the brain and the rest of the body associated with the attention and motor control. It contains the nuclei of many cranial nerves. You see this part in the double side, of course. And some cranial nerves that stem from the pons go directly to the peripheral nerve system without passing through the spinal cord. And there are four nerves here we see from the 5th to the 8th. Trigeminal nerve, vagus, it's very important nerve, nerve depressor, excitatory nerves, and hypoglossal. And the function of the pons includes sensory roles in primary information processing, the hearing, equilibrium, taste, facial sensations such as the touch and pain, roles in eye movement, facial expressions, swallowing, and even discharge of saliva and tears. And as you have already seen, here we see the nerves function, and these nerves and indicate here. And very important function contains the abducens nerve, which allows us to look from the side to side. And the very important nerves, it is here we see abduced nerves. And one more, it is the vestibular cochlear nerve, which allows us to hear. Here we see cochlea and cochlear nuclei. We do not forget about the tracts. It contains all tracts. Here we see corticospinal tract, corticobulbar tract, and medial limnico-spinothalamic tract, and here we see all these tracts. So, reflex function, it contains the locus coeruleus, which is Here we see which is the main source of norepinephrine in the brain and is involved in various functions including wakefulness, response to stress and attention. And the pons has the respiratory centers, as you see on previous slide. It is a reflex for change the phase of breathing and plays a role in involuntary control of breathing. And we move into the midbrain. Next slide is the midbrain. Next part, and you see it is sometimes we have the said that mesencephalon, just mesencephalon, the name due to the development in the middle of three vesicles that arise from the neural tubes that forms the brain of developing animals, and caudale from the diencephalon, and rostrale from the pons. It's the middle part. And function in general, function connection between periphery and thalamic area, thalamus. Its substantia nigra is closely associated with the motor system pathways in the basal ganglia of this part of the brain, and the special part for dopamine production. Dopamine, it is a neurotransmitter molecule, and we will discuss about this molecule a little bit later. So two main parts we have a midbrain is tectum, posterior part, and we have two parts in the tectum, inferior colliculi and superior colliculi, and for auditory signals and visual signals. And the second part of midbrain, it's cerebral pedicule. We have the tegmentum, the second tegmentum, which we have in the brain, and crus cerebri at substantia nigra. And let's start to study all function of this part of the brain. So get to know the tectum. The primary role of the midbrain is to act as a sort of relay station for all visual and auditory system signals. And so we have the function of this part of the midbrain. It controls auditory and visual responses. And you see the locations is dorsal region of midbrain. Here we see this part. And let's see the next slide. Here we see sometimes we have the new word, it's just roof, and sometimes a lot of Latina words. And so we have the superior colliculus involved in visual processing and controls of eye movements and eye head movements if the stimulus are light stimulus. And here we saw in the position of the dog and the position of eyes and hairs, and projects to lateral nucleus to thalamus, and then to the primary visual cortex, and the inferior colliculus for auditory processing, and here head ear movements. So it starts to take head up and sound stimulus, and special nucleus in the thalamus, and to the primary auditory cortex too. So we have the special tracts for these reflexes. And next, pay attention to the function of this department. Here are the nerve centers to reflexes of orientation in the space, to auditory and visual stimuli, and the conduction of information to the thalamus, special tracts host to the spinal cord. And the next part to the midbrain is superior and inferior collicular function. We discussed about it, but for next it is the cerebral peduncle, and we have everything in the mesencephalon except the tectum, and the region includes midbrain, tegmentum, crus cerebelli, substantia nigra, and pretectum, and nerve tracts from the motor areas of the brain to the cerebral peduncle, and then to the thalamic nucleus, and start to study the function of tegmentum. It's from the Latin covering, and between the substantia nigra and cerebral aqueduct, and the crus cerebelli not considered part to the tegmentum. Sometimes we don't have this, not part of the primitive neural tube, but he grew as projection from the cerebral cortex, and parts that were inside the primitive neural tube and remained an integral part of it after complete development. The red nucleus and are considered part of the tegmentum, and we have some parts, the ventral tegmental area and red nucleus, and periaqueductal gray parts, and we start to discuss from the just general function. Here we see the general function of the tegmentum. Motor function regulates awareness and attention, and some automatic functions, and location, you know, we find it. Let's start from the first part of the tegmentum, and here I leave the links about some very important neurotransmitters. You see the dopamine and serotonin, because of this part of the brain rich of these neurotransmitters, and you see the close to substantia nigra and red nucleus, this area, and rich of dopamine and serotonin neurons. And you see the part of the two major dopamine pathways. It's very important for, especially for humans, and because you know, maybe you know, this neurotransmitters sometimes say like just hormones, and hormones of happiness, and it function part of the pleasure system, and just stimulus like some drugs activate this region. The drugs such as cocaine, you know, this, no, especially for humans, of course, but very often we use some drugs for animals too. One of the major source of incentive and behavioral motivation, emotion, security motivation, from the hearing conditioning, auto dance, and the red nucleus is the next part of the tegmentum, and function, it's primary motor function, coordination. We have the special tracts, you know, rubrospinal tracts, extrapyramidal pathways, and for humans it is less important in humans than in many other animals. In humans the corticospinal tract is dominant because of the cortex is dominant everywhere. And control of crawling of babies, controls the muscle of the shoulder and upper arm, but lower arm and hand as well. Control of arm swinging in normal walking, The rubrospinal projection receives many inputs from the contralateral cerebellum and an input from the ipsilateral motor cortex sends the efferent axons to the contralateral halves of the spinal cord. And the next part, paraqueductal ray, and the gray matter located around the cerebral aqueduct and within the tegmentum of the midbrain. And function, the tendon modulation of the pain, a role in anesthesia and role in defensive behavior and role in reproductive behavior and maternal behavior. So it is so very important for animals. All reflexes are very important for animals. So we have already discussed about all parts of midbrain instead of substantia nigra. And substantia nigra, it's Latina, black substance, and it is a place where dopamine, a lot of this neurotransmitter production in the brain, and vital role in reward and motor control, and especially for humans. And you see, if the dopamine can to produce in this area, we have some diseases, you know, these Parkinson diseases, and you see the parts of substantia nigra, and dopamine is synthesized in the dopaminergic neurons, special neurons, and here we can see these neurons in this area, black colored neurons. And function, you see, is for dopamine production, involved in reinforcement and controls voluntary movement. If the dopamine, you see, this is normal state of this substantia nigra, and when the Parkinson diseases happens. And I have some slides for this catecholamine, but I will leave the links about this neurotransmitter. Just I move on the function of dopamine. It's critical to the reward system, you know, and controls the movement, cognitive function, regulation, some prolactin secretion and motivation, and especially pleasure after food, drugs, and we know one more, the neurotransmitter with the same function, but you just read this text about this neurotransmitter. And for humans, what we have when the Parkinson diseases happen, and we will see these processes after injuring of cerebellum, and we will discuss about kinesia, rigidity, and tremor, and just I don't stop on here. And this substantia nigra and normal state in abnormal state after diseases. So tracts, here we can find in this part of the brain, we can find a lot of tracts, and motor tracts and sensory tracts too. And also we have the special nerves. We have the cranial nerves. You see these nerves, oculomotor and trochlear, and these nerves allow to just move our eyes. And one more nerve, it's not very big nerve, but they are small diameter nerves that wind around the lateral aspect of the midbrain to enter the lateral wall of the cavernous sinus. and he has stopped here. So just you see the function of these nerves move in one place and another. And next part of the brain, exactly the cerebellum. Sometimes we have the new words little brain. Is similar to the cerebellum with its two hemispheres and hardly hooded surface. It's associated with the regulation and coordination of movement, posture, balance, and even cardiac function, respiratory function, vasomotor centers. So let's start to study the cerebellum. As you see, the white matter is inside and gray matter outside of this part of the brain. And what we have in general, we have the cerebral cortex, which covers the surface of the cerebellum, is composed of three layers, and we have at first this molecular layer is relatively cellular. It's a pocket with axons of granular cells and parallel fibers, synapsin and dendrites of Purkinje neurons. The next one, the cell bodies of Purkinje cells distributed along the deep surface of the molecular layer constitute and middle Purkinje cells. Here you see this color of Purkinje cells. So here we see just the branches of the cells, Purkinje layer, and the deeper granular cell layer, densely packed nuclei of very small neurons, granular layer. And here we see the granular cells, Golgi cells, and mossy fibers. And next, neurons in the cerebral cortex have some functions. At first, the Purkinje cells, it is the neurons which produce the neurotransmitter. As you know, it is the inhibitory transmitter, and are the largest neurons in the brain, and they have extensive dendrites trees that are brought on the cortex, the first layer of the cortex. The granular cells, all the cells are neurons, of course, are the smallest neurons in the brain. They are the only excitatory neurons in the cerebral cortex, and they realize glutamate as a neurotransmitter. And basket cells, also neurons, are located in the depths of the molecular layer, and their dendrites receive excitatory synaptic input from the parallel fibers, and the axons run transversely in the volume and the inhibit balance bands of the Purkinje cells that border of excited zones, and axonal branches from the basket synaptic contacts around initial segments of the Purkinje axons are realized this gamma-aminobutyric acidic. And Golgi cells in the granular cells layer and stellate cells in the molecular layer serve as inhibitory interneurons. So the structure is very difficult. Here we see all these parts of the cells and layers of the cells of cerebellum, and even the photo very beautiful about the Purkinje cells, green colored cells, very big cells in the brain. Move on the next slide, cortical afferent fibers. Here we see two types of afferent fibers enter the cerebral cortex. Both afferent types are excitatory to the cortex, and they send excitatory collateral branches to the cerebellar nuclei before entering the cortex. The afferent fibers are mossy fibers. Mossy fibers have termination that resemble moss, and the moss ends in synapses and glomeruli with the dendrites of the granular cells, and Efferents come from all sources: spinal cord, pontine nuclei, vestibular nuclei, and nerves, except the reticular formation. It is a special place. And clumping fibers terminate by clumping the dendrites tree of the Purkinje neurons, extending it greatly via numerous synaptic contacts, and all clumping fibers come from the olivary nuclei. Like mossy fibers, these fibers send excitatory collaterals to cerebellar nuclei before terminating in the cortex. And so about functional subdivision of cerebellum. Here we see the schematic drawing of the cerebellum and showing the relationship between the anatomical and functional division of the cerebellum. We have the three main parts. Here you see the different color of these parts, and we have first very old part, archicerebellum, and paleocerebellum, and neocerebellum. And let's start to study the function of each part. And functions: the cerebellum in general regulates the following five functions of the body: muscle tone, coordination of goal-directed and spontaneous movement, posture and balance, eye movement, motor learning, and some cognitive function like language and riding a bike, professional, some motions. And each hemisphere of the cerebellum influence motor activity on the ipsilateral half of the body, and the cerebellum compares the motor plane in turn created in the cortex with motor performance and reported from the periphery, and functions to smoothen and coordinate the movements. And this is accomplished by making synaptic contacts with the brainstem, motor centers, and the cerebral hemispheres too. And we have these areas. It's new information from the source. I will leave the links to this, and you see these areas. It's very perfect about all function of the cerebellum, and you see the motor and visual information in the left hand movements, right hand movements from the different part of the right and left side of the body, and you see the rapid eye movements, divided attention, vibral fluency, auto recall, and word it's very special place. So about a little bit about in general in function of subdivision of cerebellum. It's not like this. It is a very easy drawing about motor control, execution, motor planning, and balance and eye movement. Just it is easy. And you know that we have the special places, gray matter in white matter, this nucleus inside of the body of the cerebellum. Here we see the places of this nucleus, motor nucleus of the balance, and start to discuss about the function all parts. At first we start from the archicerebellum, posterior lobe, all vestibular part. It is a format of the special nuclear lobe, a special nucleus lying on the inferior surface or in front of posterior lateral fissure. Embryologically it is the oldest part, how I said it, it receives efferent fibers from the vestibular apparatus to internal ear via vestibular cerebellar tracts. So we have the special tracts. It's considered with the equilibrium and eye movements. So we have the new one slide about function. Archicerebellum, it's concerned with equilibrium, floccular nodular lobe, and has connection with the vestibular, reticular nuclei and brainstem. And efferent vestibular fibers pass from the vestibular nuclei in pons and medulla to the cortex, ipsilateral, nodular lobe, and efferent cortical, so Purkinje cells fibers project to nucleus, which project to vestibular nuclei and reticular formation, and it affects the special system, limbal system, bilaterally via descending vestibulospinal and reticulospinal tracts. So we have these tracts and the connection with the cortex and all these parts. And new one part, paleocerebellum, and spinal part, formed in middle line worms surrounding paravermis, and to have some parts nuclear. It receives efferent proprioceptive impulses from the tendons and from the muscles, and via spinocerebellar tracts, dorsal and ventral part. It sends efferents to red nucleus in midbrain. It's considered with a muscle tone. And next one slide about this function of this part. So in general it is a muscle tone and posture, and we have the efferent and efferent fibers in special tracts. And one more part of the cerebellum, it's neocerebellum, or cerebral part. It's the remaining largest part of the cerebellum, new ones. It includes the most of two cerebellar hemispheres and dentate nuclei. It receives efferent impulses from the cerebral cortex, pons via cerebro-pontocerebral pathways, and it sends efferents to the nucleus of thalamic, and it controls voluntary movements, especially for humans, and muscle coordination, some reflexes. It's in general the muscular coordination. It receives efferents tracts and efferents tracts too, you see, and connection of neocerebellum with the different parts of the cortex, you see here. And in general this scheme has the different ways and different connection with the nucleus inside the brain, and what stimulus we need to control these areas. Here we see the more information on this slide, and connection even with the spinal cord, and finally common pathways. And what we have the next the symptoms of damage of cerebral activity. And we discussed about ataxia, and maybe these names, and maybe you hear that some triads, triada Luciani, and one it feels these triads, triada Luciani and Charcot's triads of this integration, all dysfunction of cerebellar And we will see the video for dog with these diseases. And for end we have this picture with the damaged area of cerebellar for dog. And what we have in general, we discuss about a lot of area in a brain, and next time we start to study the encephalon. But now we have a little bit of our work. It is just work 32 in your practical book, and studying of the cerebellum function for everyone. We just have the type of experiment, and you just try to do this experiment by yourself. But we have just video for dog with the damage of the cerebellum. But for you, for your homework in your book of animal physiology from genes to organism, you should read the pages about nerve system. And on our page in a Moodle we have a little bit video files, and of course links to this practical. So it's almost all. Just let's start watch the video. Here we see the canine cerebellar ataxia, the destroying the function of the cerebellar. Let's look on the limb of this dog and the movement in general. So it is all for today and for this practice, and I hope we will see a lot of new information, and see you next time. Bye bye.